primer lugar, para mí es un placer presentar la primera charla de hoy. La primera charla de hoy la va a realizar Isabelle Masneuf. Isabelle Masneuf, actualmente ella es profesora, es como catedrática, diríamos, de Enología en el Instituto de las Ciencias de la Viña y du Vin, está en Bordeaux, es un instituto de investigación que está en Bordeaux, aunque ella formalmente pertenece a Bordeaux Sciences Agro. Ella es enóloga, tiene un doctorado en ampelografía y enología por la Universidad de Burdeos y ha trabajado toda su vida en temas de microbiología, tanto en vino como en viña últimamente también. Y nos va a dar una charla un poco de la microbiota desde la viña hasta la uva. ¿De acuerdo? Isabel, quand tu veux, la charla va a ser en inglés, lo digo porque afinéis la oreja.
Yes, thank you. I think we can start now. No, it's okay. So, the aim of my talk will be to give you an overview on the microbiota from vine to wine and what we know at that time on their genetic diversity. So as you know, microorganisms play a key role during the winemaking process. Everything starting at the grapeberry surface, where we have three main different families of microbiota, namely yeast, bacteria, and fungi. Fungi often, of course, always associated with a decrease of crop quality, crop yield. Then, during the alcoholic fermentation, we move to another system where, of course, yeast will be dominant, especially one key species, which is Saccharomyces cerevisiae, with very high population level ranging from 100 to 10 to 100 million of cells per milliliter. Then we have also a part of Saccharomyces cerevisiae, which is a dominant species, non-Saccharomyces yeast at a lower population level. And of course, also lactic and acetic acid bacteria. And we will see later that at the beginning, during the alcoholic fermentation, bacteria population is quite low, and because of the development and the growth of yeast. Then we're going on with the process, finally reach wine, and we have the malolactic fermentation, often conducted for red wine vinification, where lactic acid bacteria play a key role, especially one species which is Enococcus oeni. Again, reaching high population level of 1 million, 10 million of cells per milliliter. And sometimes, even often, occur during the wine aging process some spoilage microorganisms. One main spoilage is a yeast species, which is Brettanomyces bruxellensis. I will give you a few words on that species later. And of course, we can have also acetic acid bacteria. So, I will start from the vineyard, showing you the fact that this microbiota could have temporal and spatial variations that are very important. So everything started, we can start from the winter period in the soil, where we have also a specific microbial compartment. I think there will be some lecture on that later on. So soil microbiota play a major role, of course, in biogeochemical cycles, soil fertility and chemistry, and microbiota play a role in nitrogen fixation, phosphate, iron solubilization, production of hormones, enzymes responsible of involved in plant growth, and of course carbon cycling. So then, at the interface of vineyard soil, so yes, at the interface of the plant and soil, there is one very important compartment, which is the rhizosphere. Rhizosphere is where there is a strong interaction between soil microbiota and vine and roots. And there is some migration phenomena from rhizosphere to the above ground organs. And recently some authors reported evidence that some fermentative microorganisms, could be bacteria, maybe could be yeast, could become endophytes in grapeberries through the xylem roots, and so be part of the microbial community at the grapeberry surface of ripened fruit, of the ripened fruit. There is also some evidence that during the winter period, soil is a kind of primary reservoir for yeast and bacteria that could, so those kind of fermentative microorganisms could survive into the soil during the winter period and then will be disseminated to the above ground organs during the vegetative growth of the vine. So another very important compartment for microbiota is phyllosphere. And atmospheric microbiota is another contributor to the communities of the above organs, namely flowers, but fruits and leaves. And these environmental microbiota, they are disseminated in the vineyards through different vectors. So we can consider wind, but also arthropods like bees, wasps, fruit flies, as well as birds that could be vectors of yeast and bacteria in the vineyard. So those vectors play an important role on the colonization of, for example, leaves and grapeberry surface at ripened stage. And insects are a link between surrounding area close to vineyards, for example, forests, trees, and they could be vectors of microbiota from surrounding area and vineyards. So usually we consider that arthropods, insects, could disseminate microbiota to short distance, few meters, 1 kilometer, for example, local dissemination, whereas birds could disseminate yeast to a much longer distance, few kilometers, could be even 100 kilometers. It was shown, for example, in New Zealand vineyards. So this dissemination aspect of microbiota through different vectors is very important to understand the whole system. So there is a very important theory proposed by plant science scientists who consider the holobionts. So holobionts is a plant with its associated microbial community, and it's considered as a single entity. So the idea behind this concept is the fact that microbiota will provide new what we name phenotypes. We call these extended phenotypes. So microbial community will provide new properties, new traits that could play an important role, for example, in plant growth or in plant health, and also in biotic or abiotic stress management. And when we consider the evolution, the history of evolution of vine, we can consider that it's a unit of selection where plant and microbiome interactions have co-evolved to maintain host functionality and fitness over this evolutionary time scale. So we should consider not only vine and grapeberry, but also its associated microbiota, which is a system that work together and evolve also together. So we have seen the variation, the temporal evolution of microbiota from winter, for example, to during the vegetative growth. Then we have also evidence of temporal variation of microbiota. And the first evidence of what we name microbial biogeography was provided by a research team from the University of Davis, Nicolas Bokulich, in 2014. So this research team, they managed to collect around 270 grape must samples in the vineyards of California. And they used next generation sequencing technologies that are based on the sequence of DNA of microbial community. So these new technologies allow to gain deeper insight in biodiversity, the structure of microbial communities. And they show for the first time that the grapeberry microbial biogeography is non-randomly associated with regional vital climatic factors across multi-scale viticultural zones. So you have here the title of their publication. That is to say, when you consider different wine production zone, for example, in California, we will find significant difference in terms of microbial diversity and also microbial structure. And again, they showed that there is an impact on the variety and also an impact of climatic factors, as they shown that from one vintage to another, there was significant difference in the microbial community present in grape must samples. So then, going on with this first research work, many studies have been done at all over the world that confirm the existence of microbial biogeography. So those studies were conducted at different scales, global, regional, and local scales, taking into account different compartments. So you can see, for example, in South America, Argentina, Chile, soil roots, grapeberry leaves, and also in Italy, grapeberry bark, in Greece, grape juice, in China, grapeberry, and also in New Zealand, grapeberry. So all those scientific studies were conclude to the existence of microbial biogeography. That is to say, considering different scale, regional, local, we can find significant difference concerning the microbial community in the vineyard. So those results concern the study of microbiota at the community level, so considering a whole assembly of different microorganisms. We can also study microbial biogeography at the population level, that is to say, considering only one key species. It was done for, of course, this very important species for winemaking process. It was done for Saccharomyces cerevisiae. Many research work was done to study the genetic diversity of Saccharomyces cerevisiae in the vineyard, but also during the alcoholic fermentation of spontaneous alcoholic fermentation of grape juice. And till now, research work were able to show that there is population differentiation of Saccharomyces cerevisiae at global and regional scale, but not at vineyard scale, not at very small and local scale. So probably it's due to this aspect of dissemination, as mentioned before, the fact that, of course, considering different vineyards, you don't have physical barriers. And yeast and bacteria could be disseminated by insects, arthropods, wind, and birds. So as a consequence, at local, very local scale, vineyard scale, there is no significant differentiation related to Saccharomyces cerevisiae. Another very important aspect is the terroirist. That is something important for the wine industry or raise question. And here I consider terroirist with the fact that you have the presence of a native yeast strain in a given vineyard, which is only present in this area. So a kind of story that some specific strain will be unique to this vineyard, to this cellar. Until now, we don't have a strong scientific evidence of that. Okay, so terroirist is still an open question, but till now we don't have scientific data that will support this idea of the existence of terroirist. Again, on this story of terroirist, here I show you some results related. We were able to study the diversity of Saccharomyces cerevisiae collected from spontaneous fermentation in the same cellar for long term. So we started it was the first sampling was in 1992, 1993, and then we're going on with different vintages. We are still going on to study the genetic diversity of Saccharomyces cerevisiae population. So as you can see in this slide, in this graph, each color is representative of one vintage. So you can see that we have a specific differentiation of Saccharomyces cerevisiae population according to the vintage, where we have here in blue, we have strains collected in 2014. On the upper part, you have in pink strain collected in 2016, so quite different. And also here in green, strains collected in 2017. So this result done in a specific vineyard and cellar in Bordeaux area show you that there is no interannual stability of Saccharomyces cerevisiae population in a given winery. So time is one factor that will shape the population of Cerevisiae. So contrary to other research work showing that from one vintage to another, there could be the same population of Cerevisiae, but when you consider the long-term study, it's not the same. And in that sense, conducting long-term studies could be very important to have a global view on the genetic, the interannual stability of Saccharomyces cerevisiae population in a given winery. So what about another very important fermentative species, which is Enococcus oeni? So here I show you some very nice data obtained by my colleague Patrick Lucas at ISVV. So Patrick, with a PhD student, collected different Enococcus oeni strains from different vineyards. You can see Bordeaux, Burgundy, the south of France, but also Lebanon, and also different fermentative products like cider, but also kombucha, which is a fermented tea. So as you can see, they established this, what we name phylogenetic tree. This is a kind of representation that highlights the links, genetic relationship between the different strains. So as you can see, we have in the same branch, in the red wine, gathering isolates from wine, we have a mix between isolates from Bordeaux, from Burgundy, from Lebanon. But in the country, we have specific branch gathering isolates from cider, but also from grape juice and from kombucha. So just the idea to retain with this work is the fact that substrate, but not regions, explains the population structure of Enococcus oeni. So for this species, no evidence of biogeography existence, okay? It's more related to the origin of the substrate from which the strain were collected, probably in relation with some specific adaptation properties of the strain to cider, which are not the same as specific adaptation properties to wine and to kombucha, for example. So for Enococcus oeni, lactic acid bacteria, no evidence of the existence of biogeography. So if we move from microbiota from vineyard to wine, So at maturity stage, you know that grapeberry is a primary source of fermentative microorganisms. And when we move from grapeberry, we crush the berries, we obtain the grape juice, we will have a strong decrease of the microbial diversity due to the fact that we move from a very poor medium in contact with oxygen, which is the grapeberry surface, to another stressful environment, the grape juice, where you have high amount of sugar, you have low acidity, low pH, maybe not this vintage, but anyway. And also you protect the juice from oxidation, and you can add sulfur dioxide. So due to this strong modification of your mediums, there is a strong decrease of the microbial diversity, and only some specific species of yeast, of lactic acid bacteria will be able to survive and initiate a growth. I didn't mention before, but something very important to understand is Saccharomyces cerevisiae and Enococcus oeni is quite rare in the vineyard. If you want to collect Saccharomyces cerevisiae in the vineyard, for example, at the grapeberry surface, you should use enrichment procedures to be sure to be able to isolate some colonies of Saccharomyces cerevisiae. So both fermentative species are quite rare in the vineyard. They are not well adapted to the environment. Their specific niche is really fermentative process and fermentative beverages. So taking into account this strong modification of the medium, we should consider that vineyard microbiota is quite different from fermentative microbiota due to this selection and decrease of diversity. So we have evidence, as I said before, that grapes is the first source of fermentative microorganisms, including Saccharomyces cerevisiae. So in this research work, we were able to quantify the flux between grapes and grape juice regarding Saccharomyces cerevisiae. So we infer genetic exchange between vineyard grapes, grapeberries and grape juice. And we show that there is a strong flux of strains. You see 195, which is number of migrants per generation of yeast, from grapes to cellars. But we evidence also that there is, thank you, there is a flux back, which is less. It's 55 number of migrants per generation. So it's a kind of continuum from grapes to wine. So grapes will inoculate grape juice, and grape juice and fermentative cellars will also inoculate the vineyard. Okay, and this inoculation back will be could be through aeration system to evacuate CO2, for example, in the cellar, could be through vectors, Drosophila, for example. When you collect the pomace at the outside of the cellar, if it's close to the vineyard, you can have some flux of microbiota of pomace that could go back to the vineyards. So it's a kind of cycle, you see, this system. So in the cellar, I mentioned before that Saccharomyces cerevisiae is really the major wine species. It will dominate the system. Some authors showed that cerevisiae will construct its own niche by producing ethanol and increase temperature. And so that will provide to cerevisiae some fitness advantage over the other members of the community. That's one main reason why Saccharomyces cerevisiae will be dominant during the alcoholic fermentation of grape juice wherever over the world where we are producing wine. So when we consider the dynamic of yeast population during the alcoholic fermentation, here you have one example of spontaneous fermentation of white grape juice. It was a Chardonnay in New Zealand. So you have the start of alcoholic fermentation. We follow in blue the bricks. And you see before the start of alcoholic fermentation, we have some dominant non-Saccharomyces species, especially Enseniaspora uvarum, Candida zamplinina, which is now named Starmerella bacillaris, and some minor non-Sac species, Isachienka, Menchnikovia, and Tauraspora delbrueckii. As you can see, the population level of Saccharomyces cerevisiae is quite low. It's in green here. It started from 1,000 cells per milliliter, then reached, as I said, 100 million of cells per milliliter in few days. So as soon as cerevisiae is dominant in the system, you will have the decrease of non-Sac yeast, and even we will not be able to detect non-Sac yeast considering our methodology of analysis. So this dynamic is quite common, again, for spontaneous fermentation all over the world. And of course, one question could be, what is the diversity of Saccharomyces cerevisiae strains that conduct the alcoholic fermentation? So for that, we can sample the grape juice, the fermented grape juice at mid-fermentation, and then in the lab we will isolate some colonies. And what can we obtain? So in that case, we obtain a very high genetic diversity of the colonies, so by using PCR molecular tools to obtain a fingerprint, genetic fingerprints. And you can see in that situation, for this spontaneous fermentation in New Zealand, we obtain a huge diversity of strain of Saccharomyces cerevisiae. But sometimes we can have a dominant strain, a spontaneous dominant strain. So considering spontaneous fermentation, we can have both systems, high genetic diversity or low genetic diversity of Saccharomyces cerevisiae. Of course, when we inoculate the grape juice, we have the second situation. So we have the dominance of one strain of Saccharomyces cerevisiae. So when we add selected yeast to conduct the alcoholic fermentation, we just mimic what the natural spontaneous system can offer, in fact. So now, considering lactic acid bacteria, we have very low population level at the beginning of the process, then a decrease during the alcoholic fermentation, and then Enococcus oeni, after the alcoholic fermentation, will start to initiate a growth, reaching 1 million of cells per milliliter, and then the malolactic fermentation will start. So we can also ask the question, what is the diversity of Enococcus oeni strains during the malolactic fermentation? And I show you again some results from my colleague Patrick Lucas, who showed that, generally speaking, the number of strains during malolactic fermentation can range from 1 to 11, and in around 40% of cases, you will find 3 to 4 strains during the malolactic fermentation, spontaneous, indigenous strains of lactic acid bacteria, of Enococcus oeni. So considering the diversity of sporage yeast, we have now some evidence because we were able to develop genetic tools to decipher the genetic diversity at strain level of Brettanomyces bruxellensis. So we showed that we could have the same clones, the same genetic profiles in different wineries in a given wine-producing region. One also very specific point is the fact that we can have some clones, some genetic profiles that could be evidence over decades from wines in the same winery. We were able to collect strain of Brettanomyces in very old vintages, and so we could consider a very long period of studies, and we show that the same genetic profile were identified, were demonstrated in very old vintage, but also in more recent vintage in a given winery. So this result showed the high capacity of this species to be resident in the winery, probably through different abilities to bioadhere, to develop under biofilm in the equipment of the cellar. And now there is a kind of consensus all over the scientific community in the world that cellar is the main source of contamination of grape juice by Brettanomyces bruxellensis. So winemakers will guide the winemaking process step by step. So they can use sulfur dioxide, which has antimicrobial effects, just at vatting stage, but then after the alcoholic fermentation or malolactic fermentation, and of course during the wine aging process. Some problem with sulfur dioxide use, for example, against Brett, because we have evidence now that there are some strains of Brettanomyces bruxellensis that are tolerant or resistant to sulfites. And in that case, it's not anymore useful to increase the amount of sulfites during the wine aging process, because you have strains that could survive and even grow and produce volatile phenols, considering a high amount of SO2. So another way for winemakers to monitor the microbial population is, of course, to control temperature, which is a very important aspect. During the fermentation process, but also during the wine aging process, it's better to, during the wine aging process, to decrease temperature. And another aspect is to either use spontaneous fermentation or inoculating fermentation. So just to focus on what is the difference between inoculated and spontaneous fermentation. As I mentioned before, when you are using active dry yeast, for example, to inoculate the grape juice, you will have a very low Saccharomyces cerevisiae diversity because you will impose into the grape juice a dominant strain. We will have this situation. Whereas considering spontaneous fermentation, as I mentioned before, you can have either dominant spontaneous strain of Cerevisiae, well adapted to the grape juice, for example, or we can have a huge diversity of different strains. So usually there is one question that could be asked at that time. So what is the link between yeast diversity and wine quality and complexity? Because often I can have a look in the scientific literatures or in some websites that always when we are talking about yeast diversity could be non-Sac yeast, Saccharomyces cerevisiae, could be different strain of Saccharomyces cerevisiae that will increase the wine quality. That's better now. And also that will increase the wine complexity. So I must say that till now, from scientific literature, we don't have evidence that there is a link between yeast diversity and wine quality and complexity. So this is two different concepts, and wine complexity is not so easy to define because it's not only related to the addition of many compounds. It's because we have evidence now that there is some interaction, sensory interaction of different volatile compounds. For example, you have also interaction between different yeast, non-Sac yeast, and Saccharomyces cerevisiae. So be careful with this concept that shows that it's easy and logical to think that yeast diversity is related to wine complexity, but till now we don't have clear explanation of that, clear scientific evidence of that. One thing we know that from empirical observation, it's the fact that when you have high genetic diversity for Saccharomyces cerevisiae strain during the alcoholic fermentation, sometimes could lead to sluggish fermentations, probably due to interaction between all the strains that could develop during the alcoholic fermentation for nutrients, for colonization of the medium, and as a result, we have a slow fermentation, could be sluggish and stuck fermentations. So, just a few small take-home message for you. We have seen that microbiota plays a major role in plant health and growth, but I think this aspect will be developed during this conference. We can now consider the holobiont, that is a vine, the plant with its associated microbiota. Microbiota are highly disseminated in the environment. We have seen also that vineyards and cellars are two ecosystems with very contrasting and seasonal characteristics, for sure, but they are tightly connected. And not only on the flux, vineyard or grapes will inoculate the cellar, the grape juice, but also flux back. There could be some flux back from cellars into the vineyards. Vineyard microbiota are different from fermentative microbiota. And fermentative species are very rare in the vineyard because they are not well adapted to this environment, to this ecosystem. Be careful between the link between yeast diversity and wine complexity. It's not so evident. We can have huge wine complexity with a dominant strain of Cerevisier because you have high qualitative grape berries, very nice terroir expression. So be careful with that. And also, we have no evidence of microbial biogeography at community level. But for fermentative species, it's not so evident. And the existence of terroir yeast and bacteria is still an open debate. So thank you very much for your attention.